Hello, good afternoon, everyone. We're gonna try to finish lecture 9 today. There are quite a bit of stuff to go through, so let's get right into it. Okay, first thing first. Um, we have plasmid, right? Actually, let's do a quick recap. We were talking about how to insert a gene into a bacteria so that the bacteria is gonna transcribe and translate the gene for us and make the product. And specifically, we use the case of insulin, right, where we want to put the insulin gene into the bacteria and therefore turning it into an insulin factory, right, which would be useful because insulin is something that is needed for people with type 1 diabetes. Right, so the steps would be to locate a gene of interest using the online human genome browser. We're going to clone the gene using PCR. And now the next step is to put that gene that we've made, millions of copies using PCR, and then we're going to try to put it into the bacteria. So here is a generic bacterium, and it has its core bacterial DNA. And in addition, there are also circular DNA called plasmids. We previously talked about plasmids being able to give bacteria certain special properties. It can give them antibiotic resistance, for example, or it might allow them to grow an extra protective capsule around them so that it allows them to survive a little bit better in harsh environments. So, you know, generally, bacteria can exchange plasmids with each other, or they can pick it up from the surrounding. Let's say one bacterium dies and then it breaks down and its plasmid is just floating around in the environment, and other bacterium can come along and pick up that plasmid directly. Okay? Now, um, before we talk more about the plasmids, we need to learn a new thing called restriction enzymes. Restriction enzymes are normally found in bacteria, and their job is to protect the bacteria from external DNAs, such as viral DNA. We learned about bacteriophage before. It lands on the bacteria and injects the DNA into the bacteria. And with restriction enzymes, the bacteria will be able to shred or cut the incoming DNA. Right? So that prevents the virus from hijacking the bacteria, right? So that's what restriction enzymes are. What we can do is to actually use the properties of some of these restriction enzymes to help us insert our gene of interest into a plasmid. And then once the gene goes into the plasmid, we can feed the plasmid to the bacteria. Okay? Now, the restriction enzymes do not cut at random locations. In fact, there are specific recognition sites that are usually 4 to 8 base pair long that will be recognized by the restriction enzymes, and they will only cut at those specific sites. These sites are generally characterized by complementary palindromic sequence. What does that mean? In English, a palindrome is a word that spells the same forward and backward. So madam spells the same M-A-D-A-M forward as it does backward. If you read the letters backwards, it spells the same thing. Level, wow, kayak, these are all palindromes. And then this sentence is also palindrome. If you read it forward, it's the same thing as when you read it backwards. In the context of restriction enzymes, the enzymes will find specific complementary palindromic sequence, such as the orange sequence here. This restriction enzyme is called eCore R1 because it is taken from E. coli, and it will recognize GAATTC. This is a palindrome because if you read it forward on one strand, GAATTC, it is the same thing in the other direction on the other strand. Okay, GAATTC. Once the restriction enzyme finds the particular palindrome, it will cut it, and there are different ways it can cut the sequence. This particular enzyme will cut it zigzag, and then you will have two fragments. Okay. Now these things that are hanging over each out like that, we call them sticky ends. Sticky ends. And you can see the part that are hanging out on each of the sticky end is actually complementary to each other. The AATT here is complementary to the TTAA here. So it's possible they're called sticky ends because it's possible for them to stick back together, right? And it's not surprising because once again, they are complementary. To recap, bacteria have restriction enzymes. Different bacteria makes different restriction enzymes. They are there to protect the bacteria from outside DNA, like a virus injecting DNA into it. We, like the scientists, they take the restriction enzymes out of the bacteria, and I'm going to tell you how they will use the properties of restriction enzymes to put a gene into a plasmid, then feed the plasmid to the bacteria. That's what we're trying to do. Okay? Humans do not have restriction enzymes, only bacteria do. Okay? And we are trying to understand how to use a restriction enzyme for the purpose of turning bacteria into insulin factories. Questions before we move on? It's really difficult, today's lesson, okay? Probably one of the most difficult ones. So just ask questions if you have any. This is the table with some restriction enzymes. Okay, these are not the only ones. There are many different restriction enzymes out there. And it tells you what bacteria they are taken from. Equal R1 is taken from E. coli, right? And then you have probably, these are not very common. People might not have heard of it. You might have heard of this one. It causes like a type of influenza. But the point is, each restriction enzyme will have a specific recognition site. Equal R1 will only cut GAATTC. Small one will only cut GGGCCC, right? If you have this particular sequence and then you mix it with small one, nothing will happen. Okay, so they are very specific in terms of what they want to cut. Some restriction enzymes will cut zigzag. The red arrow tells you how it is being cut. Right, so here it will cut zigzag, creating sticky ends. Right? So sticky ends, sticky ends, sticky ends. Only sticky ends that are created with the same restriction enzymes will be able to stick together because it's called the overhang. Like the overhang section are complementary to each other. You cannot take this sticky end created with equal R1 and try to stick it with this end created with Hindi3. Okay? They're not compatible. The AATT is not compatible with the TCGA. If you create a sticky end with Hindi3 on one end, you will have to stick it with another end created using Hindi3. Okay? The 5 to 3 doesn't really matter because it's palindrome. So if you read it this way, same as if you read it that way. Okay. Now, other restriction enzymes create straight cuts. No sticky ends for these ones. Okay, we call them blunt ends. Right? for our purpose. What is our purpose again? I just want to, throw that out there and see if you're with me. What are we trying to do with the restriction enzymes again? I told you two times already. I just want to hear you say it. What are we doing here? Yes, we're trying to hijack the bacteria. That is a great way of putting it. And by that, we mean that we're trying to put the gene of interest, the insulin gene, into a plasmid, right? So in order to do that, the blunt ends enzymes are not very useful for us. Okay, we will only be using the restriction enzymes that create sticky ends. All right, so let me write that down for you. These ones are useful for inserting a gene into a plasmid. Plasmid are these tiny circular DNA that bacteria can pick up from the surrounding and incorporate it into its own genetic materials. All right, let's do some practice. We have a restriction enzyme here, small one, and it recognizes GGGCCC. Did you say direction? Oh yeah, direction does not matter. Well, I mean it kind of does. Okay, let's do this example and maybe it will help you understand more. Okay, so look, I have just a piece of DNA here. I'm gonna mix it with SMA1. Okay, like if this is hard for you to visualize in terms of like what do you mean you mix it, right? Actually, it looks like this, okay? The DNA is going to be in a very small tube, no bigger than what I show here, okay? Very small. And then the restriction enzymes also going to be in a very small tube. All right, so normally what you do is you take a pipette, you take out tiny drop of the enzyme, and then you're gonna add it to the DNA. That's what's gonna happen. Okay, you're gonna mix them up. Then the small one will look for GGGCCC and cut it. All right. So starting from the 5' end, we are looking for GGGCCC, which means I'm not gonna start looking here. I'm gonna start looking here. That I guess in a way that's what it means by, you have to do the right direction. But like it's not gonna matter in terms of whether it cuts it or not. So we're looking for GGGCCC. Okay, so let's just go through it, and there is the palindrome, GGGCCC. And if you read it the other way around, GGGCCC, it's the same thing. That's a recognition site for a small one, and it's gonna chop it right down the middle like that. Is it going to cut at an other location? Okay, if you keep looking, you will find an other GGGCCC, right? That's an other recognition site, right? Again, 5 to 3, GGGCCC, and that's where it's gonna do another cut. So after the cuts, right? This is one piece, two piece, three piece. If you take a piece of paper and then you cut twice, you have three pieces, right? Three fragments, we say. All right. Next, identify palindromic sequences. Okay, so there are a few here. I'm just gonna highlight 2 or 3 for you to see. For example, over here, the palindromic sequence has to be at least 4 nucleotides long, right? So that's a palindrome. It's hard to tell on its own, but if you write the complementary strand using base pairing rule, you will see that it is a palindrome. GCGC. If you read it backwards, it's also GCGC. Okay. It's not on the same strand. Like that, that's not a palindrome. Even though forward is TTT and backward is TTT, that's not what we're talking about. We're talking about complementary palindromic sequence, which means it's like one strand versus the other strand. If they read the same in opposite direction, then it's a palindrome. Okay. Here is another one. Like for you, it might be easier to write the whole thing out and then look for it. Okay, but there you go, GAATTC. If you read it backwards, it's the same thing. Okay, and then there is another one here. CTTT, AAAG, CTTT, AAAG. Okay, so there you have it. Three palindromes. There might be more. You can look for them, but hopefully you understand the three that I have chosen here. Let's do another question. Okay, I'll give you a moment to try this. We have a piece of DNA here. Remember, DNA has two strands. And we are going to treat it with two restriction enzymes, EcoR1 and HIN-D3. That's how you pronounce it, okay? HIN, and then the D, and then that's Roman numeral 3, HIN-D3. And then you have the recognition site at the bottom, and please go ahead and try to see how many times the fragment will get cut. How many cuts? Anybody? Okay, I got some answers coming in. Very good. So let's take a look at equal R1. Equal R1 is GAAATTC, and it's in the 5 to 3 direction. So I'm gonna start looking at this top strand. So GAATTC. There we go. Right off the bat, we have a GAATTC. And remember, the red arrow tells you how it's going to be cut. So that's one cut right there. If you keep on looking for another GAATTC, you will find it right here. Right? That's another GAATTC. So that's an E core R1 cut and other E core R1 cut. And then the HIN-D3 is AAG CTT. So AAG CTT is right here. And we're going to have another cut right there. Okay. So together, one cut, two cuts, three cuts, four fragments. One fragment, two fragments, three fragments, four fragments. If you take a piece of paper, like a strand of paper, do it yourself if you have a hard time visualizing. Okay. Count it 3 times, 4 pieces. Everybody okay? Before we go to the crazy notes, which is going to take a long time to write and even longer to explain. All right, here we go. Steel yourself and brace for this next section. Okay. So this is about inserting a gene into a plasmid. We'll do that first, and then we're gonna feed the plasmid to the bacteria. All right? Okay. So you can buy plasmids online. Oh, the program just hang. Just give me a second, I gotta restart. Okay, let's see if it works now. There we go. Okay, so this is a plasmid, and yes, bacteria has plasmid, but like biotech companies also make their own plasmids. I'll tell you more about it in a second. Okay, when you buy a plasmid from companies, it will give you a map that tells you everything about the plasmid. Let me see if I can show you a plasmid map, just so you have an idea of what it might look like. Plasmid map image. Okay, there we go. Look, right? That looks very complicated. Right here. That's the picture. And that's what a plasmid map looks like. Okay, like that's a simplified version. It has all these things on it. And then, it tells you the exact sequence in some cases. Um, so that it's very complicated, basically. But we're gonna simplify it, and I'm gonna say in this particular plasmid, right, there is a sequence, and the sequence is TTA. Let's do TTTAAA. I'm just making this up. This is hypothetical. TTTAAA. Okay, so this plasmid has a recognition site. Recognition site for restriction enzyme X. Okay? It's just a random restriction enzyme I make up. Restriction enzyme X. That means when you treat this plasmid with restriction enzyme X, it will cut it. Okay? And I'm gonna say it's gonna cut it like that. Let me use a different color here. It's going to cut it right there. Okay, it's gonna do a zigzag and basically pops the plasmid open. So let's see what that looks like. Okay, we're gonna add restriction enzyme X, and afterwards, this guy's gonna pop open, and this is the part where I always messed up. Let me just try. Don't copy yet until I tell you to, okay? Because I sometimes make a mistake here. All right, now you can copy. So the restriction enzyme is going to basically cut at this particular place, and there are no other recognition site in the entire plasmid, and afterwards it's going to open it up, creating, what do we call these things that are hanging out?
Sticky.
That's right. Okay? REX, the restriction enzyme X, creates complementary complementary sticky ends. They are sticky because they can stick back together. So far, Not too bad, I hope. Remember, this all happens in a tiny, tiny tube. They are called Eppendorf tubes. Usually that's where you do these experiment in. And it's not just one plasmid. We're talking about millions of these plasmid in the tiny solution. You cannot see it with your naked eye. Okay, it would just look no different than, some cloudy water, right? But it's in there, right? All these plasmid. And if you add the restriction enzyme, it will cut them open, you know, like one of those rings that you can use to uh loop your papers together, right? Okay. So where is our gene? So here is our gene. I'm going to draw a rectangle to represent our gene. Let's use purple right there. This is the gene of interest. Okay. In our example, in our case study, the gene of interest is the insulin gene. Okay. Gene of interest. That's the one you want to hijack the bacteria with, right? Now, in order for this thing to work, you need to make sure the gene that you're trying to put in are flanked by the same recognition site that exists in the plasmid. In the plasmid, we have the TTTAAA. You have to make sure whatever gene you are trying to put in the plasmid has the exact same recognition site. If it doesn't, then you have to use a different restriction enzyme. Okay, and there are so many restriction enzymes. Uh, like basically you just plug it into the computer program, and then the computer program will search the database and tell you what recognition sites are available flanking your genes. Okay, so like you don't actually have to physically do it nowadays. The computer does it for you. But you understand what I'm trying to say, right? The gene here, I'll write in black. The gene of interest. Okay, I should change that to purple so everything is nicely color coded. The gene of interest needs to be flanked. Flanked means like on both sides by the same recognition site as the one in the plasmid. Same recognition site as the one in the plasmid. The blue and the red must be the same. Okay. But not inside the gene. Okay? We only want the TTTAA to be on the flanks, but not inside the gene. If it's inside the gene, then you will cut the gene when you try to use the restriction enzymes, right? You don't want to cut the gene. You cut the insulin gene, you have no insulin, right? So only on the ends, but not within the gene. Okay? Let's say within. That's probably sound better. Not within the gene. Are you guys okay? Now remember, this thing, we already have like millions of copies, if not billions, millions of copies from PCR. Okay, so the last step is already done. They are again sitting in one of these tiny tubes. Everything happens in tiny tubes. Yeah. I've actually done these before, okay? So like I'm not just making them up. I'm speaking to you from experience, okay? So you're gonna have the genes in these tiny tubes, and then you will get another tiny tubes that has the restriction enzymes in it, and then you're gonna add the restriction enzymes to the DNA, right? So let's draw that out and see what happens. So this is gonna you're gonna mix it. You're gonna mix it with restriction enzyme X again, just like before, right? Before you mix the plasmid with restriction enzyme X, now in a separate tube, you're gonna add the restriction enzyme X to the DNA that you just cloned from PCR. And what will that do? Well, it will basically take this thing, just watch first and then copy after. I'll give you time. Okay. What's going to happen is the restriction enzymes will find the recognition site and cut it. It will cut it like that. Right? And what would you get in the end? Well, you will get this. You will get this. This is going to get cut off. Okay. And you create a sticky end here. And then over here, you're going to cut this stuff up. Okay. I think I cut a little bit too much. And then you will have a sticky end. Okay. Now this sticky end is not gonna go stick here because like it's like too narrow. It's not gonna like loop back on itself. All right, too that's like too not long enough basically. However, okay, however, this is the most important part for you to understand. I will stop after this and let you copy, okay? However, this end here, look, TTAA, right, is actually compatible with this end, right? And this end is compatible with that end. So if you mix the plasmid that you've cut open with the gene that you've cut open with the same restriction enzymes, then the gene will now put itself into the plasmid and you can glue them together, right? If you don't fully get that, that's okay. I'll draw it out for you, but copy whatever you need to first. Okay, I'll give you a minute or so. Meanwhile, let it sink in. Ask me something. Okay, so we have the plasmid and we have the gene. And basically, what you want to do is to put them together. Really? It's mandatory? I did not know that. I don't think it's mandatory. It's anonymous for sure. Um, but yeah. Anyways, so we mix together. Mix together. Okay. And then what do we get? Oh, you asking me, is it mandatory? No, 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 no. The surveys are not mandatory, all right? But if you do it, it will be very useful for us, okay? So it's a favor we're asking you, all right? Okay, let's let's see what's gonna happen. All right, so if you mix them together, it's gonna look like this. Okay, I'm not gonna write the letters because it's already very confusing. Okay, I gotta open this a little bit more. It's not the best drawing, but I hope you understand, once again, this part is gonna stick to that part, and this part is gonna stick to this part. And in the end, you will get the gene inserted into the plasmid. Now, no one could answer me this the next question this morning in my morning class. Let's see if anybody knows the answer. You can't just mix them together and expect them to be permanently glued together. There is an enzyme that we can add to glue things together. What's the name of the enzyme? We learn it in lecture 6. Yes, the ligase. Right, so you're gonna add a little bit of ligase as well, which will help seal the deal. And now we will have a bunch of plasmids with the gene of interest inserted into them. There we go. Any question? What do we want to do with these things? What's the next step here in this experiment? What are we trying to do with these plasmid with the gene of interest inserted? That's right. The next thing we're going to do is we're going to feed them to the bacteria and encourage them to take it up. Okay, you cannot physically inject the plasmid into the bacteria. Okay, that's not possible. You can't like look at them under the microscope and then everybody gets a shot or something like that. It doesn't work like that. You can expose the bacteria to the plasmid, and you can use different methods to encourage them to take it up. Okay, so that's what we're gonna do next. But before that, seriously, are we together? Remember, this is gonna be on your quiz next week, right? Um, so make sure you go through it. Holding on, yeah. Okay, thank you. All right, let's take a look at the plasmid here. All right, I'm gonna explain the different features of the plasmid to you. Why we use a plasmid? Yeah, because bacteria naturally will have a tendency to take up plasmids, right? So it's not something that's completely foreign, right? If you use something that is like totally uh like foreign to a bacteria, then there's a very low chance that they will take it up, right? Because how bacteria behave, they would take up plasmid randomly on their own anyways. So it makes sense for us to provide them with a plasmid, hoping that they will take it up. Does that make sense? Okay. I just think of a story. It's like, when my kids were younger and, they need to take medications, right? Like if you ever try to feed a kid medications, like I don't care if you use like bubble gum flavor, banana flavors, honey flavor, whatever, like they just won't eat it, right? If you give them straight up, they will refuse. Okay. So you try to mix it with things that they normally would eat, like, ice cream or something like that. Right? So like you mix the ice cream with the medicine and then you feed it to them. And hopefully it will go down and stay down. That's basically what we're doing. Okay. The gene of interest is the medicine, and then the plasmid is the vector. It's the ice cream, right? It's like the thing that they normally would take up, right? So you're hoping that by mixing it together, you deliver the gene silently into the bacteria. That's kind of like the situation here. Okay. Totally just uh, the analogy made up. Hopefully it helps. All right, so let's take a look at this plasmid right here. Uh, this is the name of the plasmid. Right? This is where the transcription and translation place would happen. It's the origin of it's the promoter basically. Okay, not very important. What's important are these things on the outside. Do you recognize what they are? What are these things on the outside? Right, we have equal R1, we have BAMH1, AVA1, PST1. These are
Restricted enzymes?
that's right. So these are restriction enzyme recognition sites to be more specific. Meaning, if I mix this plasmid with PST1, it will cut it right here. If you mix it with E-core R1, then it will cut it right here. If you mix it with BAMH, then it will cut it here, or it will cut it here. So on and so forth, right? So depending on what sequence flank your gene, you might need to use a different restriction enzyme. Okay. So equal R1, the recognition site is, GAATTC. But what if you don't have GAATTC flanking your genes? Or what if you have a GAATTC inside the insulin gene? Then you cannot use equal R1. Then you have to use something else like PST1. Right? So you check to see what's compatible. So when you buy a plasmid, it comes with a lot of restriction enzyme insertion site. Like all these, I know you cannot read it, but the point is you can see there's a lot of black stuff on the outside. All of those are restriction enzyme insertion sites. So it gives you versatility as to where to insert the gene. Okay, so these are restriction enzyme recognition sites. Okay, it tells you where you can insert the gene of interest. Okay, on the inside. These are called selection markers. Selection markers. Selection markers are important for you to tell whether a bacteria has accepted the plasmid or not. Okay, so let me draw this out for you. So you're gonna have another tube, another one of these tiny Eppendorf tubes, and you will have bacteria in them. And usually they use E. coli for these kind of things, but other kind of bacteria works as well. So these are bacteria in the tube, okay, and then over here you would have the recombinant plasmid is the plasmid plus the human gene in it, right? Okay, and then you're gonna mix them together. Right? You take some of the plasmid and then give it to the bacteria, and then in the end, what would you get? Right? So this is a bacterium. You get just gonna have a bunch of bacteria. Okay, and then you will have the plasmid around them. Some will take up the plasmid. Some will take up the plasmid. Okay, but actually a lot will not. Okay, you can't really force them. You can encourage them to take it. When I say encourage, like there are different conditions you can subject the bacteria to make it more likely to take it up. Like you can shock them with a bit of electricity, right? Sometimes you can shock them with cold or heat, and that will make them more likely to take up the plasmid. But it's hard to get 100% acceptance, right? So these guys accepted the plasmid. Right? They have been transformed. That's the actual name, okay? Transformed bacteria. Okay, and then these guys, they rejected, rejected the plasmid. Okay. They are, untransformed, non-transformed, whatever. So here is the problem. Everything happens in that tiny tube, right? And if you look at it, there's no way for you to tell which bacteria has accepted the plasmid and which hasn't. You just can't tell with naked eye, or like even if you put them under the microscope or something, you can't tell. They look exactly the same. Okay? That's why we need a marker, a selection marker. There are different kinds of selection marker, right? And we don't have time to go through all of them. So a selection marker is basically something in the plasmid that allows us to detect, uh, to 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 distinguish, okay, to distinguish, to tell apart, to distinguish transformed bacteria from untransformed ones. Okay. In the tube, they look the same, but as you will see, the selection markers will give us a method to tell the two apart. All right, I'm going to take a pause here, see if you want to say anything. All right, moving on. Antibiotics, such as ampicillin and tetracycline, kill bacteria. Okay, that's self-explanatory. You grow bacteria in a agar plate. We call them agar plate. I'll write down for you in a second. Okay, grow them in a dish, and then you give them nutrients. They will be happy and they grow and then they grow. But if you start adding antibiotics to the plate, then you will kill the bacteria. Again, that's self-explanatory. Okay? Let's take a look at the plasmid. The plasmid we are using in this case has two selection markers. Over here, we have the AMPR gene, and over here we have the TETR gene. What are they? The AMAR gene, sorry, the AMPR gene gives bacteria resistance to ampicillin, meaning they will be able to grow in the presence of ampicillin. Even if you add ampicillin to the solution, if they have this plasmid, it will not affect them. They are resistant to the ampicillin, but only if they have the plasmid. Tetracycline, resistant gene here, right? TETR. The TETR gene gives bacteria resistance to tetracycline. Okay? And so if the bacteria has accepted the plasmid, it will be resistant to tetracycline. And even if you add the tetracycline to the plate, it will not bother them. They will still grow. Okay? Ampicillin resistant gene only protects them from ampicillin. It does nothing in the presence of tetracycline, and then vice versa. All right, let's go over here. I am going to go through a few cases with you. Okay, let's make a big chart here and see what's going on. Sorry. It's like all over the place here. It's too much to tell you. Okay, let's just do no chart yet. Okay, let's just do a example here. All right. So let's say a gene, the insulin gene, is inserted into the plasmid with E core R1. Okay, so the gene goes right here, right? You see that? Goes right here. And then you feed it to the bacteria. I'm just gonna, you know what? Let's draw it again. And then you feed it to the bacteria. And in the end, what are you gonna get? Yeah, let me just draw a bunch of these guys. Okay, I have a bunch of bacteria here. Okay, and like I said, only some of them will have the plasmid. Okay, I'm not gonna do the purple bits. I assume you understand it by now. And like I said, you can tell just by looking at it which one has it and which one does not. And so what you need to do is you need to grow them on an agar play. Agar play. Yes, Elphys, that's correct. Okay. So this agar play, it's like the play I showed you before, right? Like in the introduction last time. Like that's called an agar play. Okay. And all these are like bacteria growing in it. Okay. So the agar play is going to have some nutrients. Okay. All the basic nutrients that you need to grow bacteria. And then basically, this is the tube that has all the bacteria, and then you're just gonna add them to the agar play. So this one has nutrients. What will the bacteria do? They're just gonna grow colonies, okay? These are bacterial colonies. They are very happy. Okay, you put this thing in a 37-degree chamber and you come back, a day later, whole thing is full of bacteria. Okay, so they're very happy. Okay? Lots of growth. Lots of growth. Okay? This is actually a control. A control to show the transformation process did not kill the bacteria. Okay, remember I said to encourage them to take up the plasmid, you gotta shock them a little bit with electricity, you gotta heat them up sometimes. These are not a friendly things to do a bacteria, and in some cases you might end up killing them all. So just to make sure that you didn't actually kill off everything, you need to put them in a control plate with no antibiotics, and you should expect them to grow. If you put the stuff into the control plate and nothing grows, you did something wrong. You kill the bacteria. Okay? When you do experiment in real life, usually it doesn't work the first time. Okay? And then sometimes you have to do it for a couple months and it might still not work. So you have to troubleshoot. You have to figure out, oh, why did the bacteria die? Right? And then you have another play, right? Another agar play. This is the selection play. Selection plate. Okay, so in this selection plate, we're gonna have nutrients. Nutrients. And then we're gonna add an antibiotic. In this case, let's add ampicillin. Ampicillin. Okay. So you're gonna take it from the same tube, okay, and then you're gonna add to the agar plate here with nutrients and ampicillin. Do you expect to have lots of growth? Or do you expect it to have no growth or little growth? What do you expect? Now, I'll tell you, and then you can practice in a bit. That's correct, Sudima. Over here, we have a mix of bacteria. Only some of them will be transformed, but some has not, right? So if they are transformed, if they has accepted the plasmid, then they will have the ampicillin-resistant gene, which allows them to grow in the presence of ampicillin. So those will grow. Okay? And anything that has not accepted the plasmid will die. Right? So only transformed bacteria will survive. Okay? Because they have the AMPR gene. Right? So if you take a look at that colony, okay, like figuratively speaking, you actually cannot see them. Okay. Like, I mean, you can see them under the microscope, but you wouldn't be able to see the plasmid. That's what I mean. All of them would have the plasmid. Okay. The fact that they are growing in the presence of ampicillin tells you they have accepted the plasmid. Okay. And that's how you distinguish the transformed ones from the non-transformed ones, right? That's why it's a selection marker, right? So similarly, you can do this thing with the other selection marker. You can add tetracycline instead of ampicillin, and you would have the same result. Are we together? Let's try this again. Here is where it can get a little bit tricky. If you are following so far with everything I said, well done, good job. It's not easy to understand this stuff. Okay. Now, did I say case one? No, I didn't. Okay, so that was case one. All right, now we're gonna do a second case. Okay, we're gonna insert a gene using. Remember, we can insert it at any of these spots. Okay, because those are the restriction enzyme sites. We're gonna insert a gene using PST1. Okay, PST1. All right, and I am gonna have 3 plates this time. I'm gonna have 3 plates. 1 plate, 2 plates, 3 plates. Okay, so all of them is gonna obviously have nutrients. Uh, I'll just write it. Okay, nutrients. Nutrient and nutrients. This one I'm gonna add ampicillin. This one I'm gonna add tetracycline. Okay? So if we insert with PST1 is going to go right here. Inserting with PST1 actually, how do you spell actually? Destroy, destroyed the ampicillin resistant gene. Okay? Because look, PST1 cuts right in the middle of the ampicillin gene, and then you will put your gene into it, which totally disrupts it. Okay? So what does it mean? Okay, so let's try to grow the bacteria in each of the plate. If you grow it in just nutrients, okay, lots of growth. Okay, lots of growth. As expected, lots of growth. That's our control. What if you add ampicillin into the mix? Will there be any growth? Some growth, no growth, lots of growth. What happens? Okay. Well, let's see. If the bacteria rejects the plasmid, if they have not taken up the plasmid, they will definitely die because of the ampicillin. Agree? No plasmid die in the presence of ampicillin for sure. But what about those that have accepted the plasmid? What about the transformed ones? Well, the transformed one will have the plasmid in them, but because we inserted a gene using PST1, the ampicillin resistant gene is actually non-functional because our gene, our purple gene, interrupts it. So despite taking up the plasmid, they will not have the resistance. And so, no growth. How about if you add tetracycline? Well, is the tetracycline gene, resistant gene intact? It is, right? Because we inserted over there and not here, so the tetracycline resistant gene is intact. And so what we're going to see is we will have some growth. Okay, some growth. Okay, and anything that's growing means uh they are the transformed one. So in this case, tetracycline is a good marker to use to distinguish the transformed versus the non-transformed. Ampicillin would be useless because everything is dead. Okay, you even killed off the ones that are transformed. Understand? All right, case three, and then we're gonna move on. Case three. What if you insert using BAMH? Okay, BAMH. BAMH1. What's an appropriate selection marker? So if you insert right here into the BAMH, sorry, into the right here using BAMH, it will go right into the tetracycline resistant gene, which means they will not have tetracycline resistance even if they take up the plasmid. So you should be using ampicillin. I don't know what color anymore. Oh, there we go. Should be ampicillin. All right? That's all. So once you've selected the transformed bacteria, you basically put them into you literally scoop them up and then you put them onto another plate and you just let them grow. And then at the same time that they're growing, they would pump out insulin because the plasmid contains the insulin gene, right? It will try to make the products on the insulin on the plasmid, and then you will purify the insulin from the culture, and that's how you make them. All right. So, I have not posted this yet, but I will. Give me a moment. I will post because it's like really hard to understand, right? So like I'll post a document on in today's announcement. Just read it. It's basically what I explained to you, and then I gave you different examples, okay, and then I give you another plasmid for you to try, and after you try it, I post the answer at the end as well. Right, so hopefully this will help you understand more. All right. Why don't we take a quick break? 5 minutes, I guess. 5 to 7 minutes. Okay, we'll come back at 1:45 and we'll do more. Okay. See you in a bit. All right, so let's keep going. Uh, Basically for the first half of the lecture of the lecture nine, we talked about recombinant DNA, right? First with the golden rice example, and then with the medicine application to hijack the bacteria to make insulin for us. And along the way, we learned a few techniques. Okay, we learned about PCR, which is used to amplify DNA. We learned about restriction enzymes, which are used to cut DNA. And then we learn about transformation and selection, which is about feeding plasmid into bacteria and causing them to make a particular protein product for us. Right? So next we're gonna learn a few other things before we finish with the lesson. We have what's called DNA fingerprinting. And you probably heard this before, or like if you watch like crime shows or something like CSI, they sometimes do these DNA fingerprinting to determine whether a suspect is at the crime scene or something, right? So as the name implies, DNA fingerprinting is it's like your fingerprints. It's unique for everybody, and you are able to basically have as little as one cell from the person, and you can um, come up with a DNA profile based on the genetic material in that cell, and you can compare the DNA profile with other profiles to see if it's the same person or not. DNA fingerprintings can also be used to determine the relationship between individuals. Like, are they siblings? Are they, parent and child relationship? And we're gonna try to look at all these examples, hopefully today. Okay. I don't think we can finish the lesson today, but that's okay. We'll do as much as we can. All right, DNA fingerprinting. To understand how DNA fingerprinting works, there is something called polymorphism. Polymorphism is a big word, but it basically means a detectable difference in the DNA of two individuals. Okay. Now, that sounds more complicated. Sorry, that sounds easier than it actually is, right? Um, if you compare your DNA and my DNA, do you remember what percentage would be identical? I told you this before. Anybody remember? Your DNA, my DNA, what percent would be identical? That's right. 99.99% would be identical because we are both humans, okay? It doesn't matter we're different race, we're not related or anything like that. We are both humans, so our core DNA is 99.99% identical, right? So finding those differences are actually trickier than it seems, right? So any detectable difference is what we call polymorphism. All right. The polymorphisms, the difference can occur in either the coding regions or the non-coding regions. It could be part of an exon, it could be part of an intron, and most of the time, these differences is as a result of mutations that has occurred over time, right? In our ancestor, we all have some common ancestors, and over time, the mutations cause us to become more and more different, okay? And that accounts for the 0.01% difference between human beings. Now, of the different types of polymorphisms, of the different types of detectable difference, one particular um, type that we are interested in is called STR, which stands for short tandem repeats. They are short repeats, sorry, they're short repeats that goes one after the other. That's what tandem means. Tandem means like lined up one after the other, right? So for example, over here we have CGA, 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 CGA. We would call this, oh, did I read that wrong? I totally did. I mean CAG, 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 right? So this would be CAG bracket 4, right? It's repeated 4 times. So it's short because it only has 3 nucleotides and it's repeated in tandem. So it turns out there are various short tandem repeats that are scattered throughout our genome. Okay? They would be on the same chromosome for everybody, but the length of the repeats would be different. Your CAG on chromosome 17, for example, may be repeated 10 times. Okay? Mine may be repeated 3 times. Okay? So different people tend to have different number of STRs, and there are many different STRs scattered throughout the genome. Okay, you might have STRs on chromosome 17, you might have, hundreds of STRs on chromosome 4, whatever, right? And since we mapped out the entire human genome with the Human Genome Project, we know exactly where these STRs are located in our genome. So, just on a side note here, there is something called Huntington disease. This is a dominant disease, right? Meaning if one of the two alleles has the mutation, then the person will get Huntington, right? Huntington is similar to other forms of neurodegenerative disease, and the person is going to slowly lose motor function, and there's no cure for it. And people who have Huntington disease tend to have the disease later on in life. By the time they have it in life, they would already um, have had kids already. Okay, so people who find out that their parents have Huntington disease, actually they have a 50% chance of having it themselves. Okay, and that's a scary thing, right? There are genetic testing available to find out whether you have it or not, but that's a really heavy question. Like if there is a 50% chance that you might have this disease because one of your parents have it, do you want to find out? Okay, some people wants to find out, right, so that they can plan, right? Let's not risk passing it on to the future generation. Like maybe they decide not to have kids. Okay, but some people don't want to find out because, like, what if you have it? right, then your outlook in life might be very grim, right? So, there are arguments on both sides. But the point is, the Huntington disease actually is it's linked to what are the effects of Huntington? It's like progressively loss of motor functions, basically. Okay, you slowly lose control of your body, and eventually you're not going to be able to move at all. Uh, where was I? Right. So this is related to one STR, okay, which is the CAG. Okay. The CAG is normally repeated 10 to 35 times within the Huntington gene, the HTT gene. Whenever these tandem repeats are copied, is common for the polymerase to make a mistake, right? Because they're all repeats, when you're trying to copy it exactly, sometimes the polymerase would slip and then it would like accidentally insert an extra copy of CAG, or it might be missing one. Right? It's like this. If I give you a whole page of T's and I ask you to copy it, it's reasonable that you miss a few or you add a few extra, right? Because everything is the same. It's really hard to keep track sometimes, right? And that's what's happening here. So if the CAG is repeated 10 times and 35 times within a gene, it's normal. The person's not going to have Huntington. But anything beyond 36, right, that's going to give the person Huntington, right? So sometimes you might not see this disease in many generations, and then all of a sudden it shows up out of nowhere. That probably means that, the CAG length has been increasing over the generations. Okay? Like maybe the previous generation, it got to 35 and it was okay. But then when the DNA was copied and passed on to the next generation, all of a sudden there's extra CAG. And then that's where it starts, right? But not all short tandem repeats, not all STR are related to diseases necessarily. I'm just giving you an example here. All right, let me try to explain this STR business with you, and then we'll call it a day. Okay, let me just see how many more things. STR analysis, and then okay. So like I said, not all STR are linked to disease, and these STRs are found in everybody. Some are longer than shorter. People who are related would have similar length, obviously, but people who are unrelated, they will have quite different um, STR length. So over here, how long is this particular STR? Well, it is 4 nucleotides and it's repeated 12 times. So 4 times 12 is 48 nucleotides long. Right? I am oversimplifying things here, okay? Obviously. So GATA is 4 and then it's repeated 15 times, so it's 60. Okay, the math is correct. Okay, I'm not simplifying the math, but like these repeat numbers are, not necessarily this small. Okay, and 4 times 30 is going to be 120. Okay, and so on and so forth. Which means everybody, if you take into account a bunch of STRs, everybody will have basically a series of different DNA lengths. Some are long, some are short, and it kind of creates a barcode that's unique for that person. Okay? And that's what DNA fingerprinting is about. It's about reading that barcode and comparing it to other barcodes and see whether they are similar, exactly the same, or completely different. If it's similar, they are probably related. Okay. If it's completely different, then it's the wrong person. If it's exactly the same, then it has to be you. Okay. DNA fingerprinting is very, very accurate. Now, to understand how this works, we have to learn about another technique. This is called gel electrophoresis. Electrophoresis. The function of gel electrophoresis is to separate DNA based on its length. Okay? Based on its length. So if you have a mixture of DNA in a sample like this one, some are 48, some are 60, some are 120, How do you separate them? You would do gel electrophoresis. Okay. So I'll try to explain to you what happens here, and I'll show you a video. We have a power supply here, which supplies an electric current. The electric current will go from negative to positive. Okay, that's the current. And in this plastic chamber is going to be a solution. The solution helps conduct the electricity. Okay, it's a buffer solution, we call it. And then inside, we're going to have a gel. Okay, so the gel is like Jello, right? It comes in a bottle of powder, and then you mix the powder with some solution, some buffer solution. Okay, the same stuff that you put in a chamber, which is mostly water, but like you would regulate the pH, okay, of the solution. Not important, right? What's important is you're gonna mix the powder with the we'll say water, and then you microwave it, and then you pour it into a cask, and then you will get a gel. Right? The gel looks like this. This is a piece of gel. It's literally like a flap of gel this size, okay, of a size of a calculator. And then there is going to be a little slots here at one side of the gel, like here. These are the slots. That's where you will load your DNA. I'll show you a video in a second. It'll make sense, okay? You load the DNA here. Okay. So this is the negative side, this is the positive side. When you run the electric current, because DNA is negatively charged, the sugar phosphate backbone, right? By now you should know phosphate is PO4 3 negative from chemistry class. So it's negatively charged to DNA. The current will push the DNA down the gel. Okay. Current will push DNA down the gel. Now, if you look at the gel at high resolution, if you zoom in with a microscope, then the gel looks like this. It's like a honeycomb structure. All right. So the honeycomb structure allows smaller pieces of DNA to move through it easily, but the longer one will get tangled up and it's not going to be able to move as easily. Okay? If you have kids, you might have seen something like this at the playground before, okay? And if you don't have kids, you might have seen it before too, I suppose. But one time I took my kids to the playground, and they challenged me. They're like, okay, let's see who can get down fastest. All right, so we all enter at the top, okay, and we try to get down to the bottom the fastest. And then in between, there are all these like tapey things that you have to weave through. So, the kids are really small. They're able to get through really quickly, right? And I get caught up along the way and it takes me a long time, right? So by the time they're at the bottom, I was just like, somewhere in the middle. And that's what's happened. That's what's going to happen here. If you have a mixture of DNA, some long, some short, the short DNA will be able to get very far in a lane. This is one lane, okay? I should tell you that. That's one lane. And then the longer DNA is going to stay closer to the top. Okay. So each well here, you will be able to put a different sample. Like you might put a sample from a person A, you might put another sample from person B, person C, and then here you might put a sample from the crime scene. And then you want to see which one was at the crime scene. So their DNA will stay in their own lane, and then it will get separated based on the length of the DNA. Okay? Now I'm going to show you a video and I'm going to explain to you how it works. That's going to be the last thing I showed you, and I cannot record that because if I record other YouTube videos in my video and then I post it on YouTube, they will say I have copyright problems and then they would take my video down. Okay, so I want you to be able to see the rest of the recording, right? So I'm going to stop the recording now. Um, After the video, it's going to be end of class, and next week is going to be the quiz on Wednesday, and come back. We'll finish lecture line and also start on number 10. So let me stop the recording right there.